This is a revision video for B5 Health and Disease, and this video is just for the triple students, so please only watch this video if you are in A1. And the first thing we're going to look at are pathogens. Now, I could talk about pathogens for 15 minutes. There are a lot that you need to know about pathogens. I'm going to try and keep it brief in this video, but if you do feel like you need to go and learn about a particular group of pathogens, please you can use Seneca, you can go on BBC Bite Size, you can email your teacher for more information, you can use the revision guides, but this is kind of the things that everybody needs to know. Okay, so bacteria. So bacteria are prokaryotes that don't have a nucleus, and they are killed by antibiotics. The first kind of bacteria infection that was discovered was by an Englishman called John Snow, and in the 1800s people didn't have running water in their houses. They had to go to wells in the town and get water. So this is a kind of map of a town, and the blue X's there are where drinking water is found, and they also didn't have running water to get rid of their waste, so they would have to go to the toilet in a bucket and empty it out into sewage pits. So cholera is a bacteria that causes sickness and diarrhea, and it can kill people because people basically can't get enough water because it just passes through them. What John Snow realised was that people that were getting cholera were getting their drinking water from a particular well, and what he concluded was that the dirty sewage was seeping into the well, and people that had cholera, it was in their feces, and it was then going into people's drinking water, so they were catching it. So this sort of experiment saved a lot of lives because they just closed that well down, and they realised that they needed to keep the drinking water wells well away from the sewage wells. So that was the first kind of evidence that a bacteria was causing these diseases, and knowing things like that saves lives. So what we'll have now are antibiotics. So even if someone gets a bacterial infection, it's fine because we can take antibiotics. So antibiotics were kind of discovered by Alexander Fleming. And one disease that used to kill lots and lots of people was TB, tuberculosis. If you got tuberculosis now, it's very, very uncommon because less people have it because of antibiotics. But if you did get it now, you could go to the doctors or the hospital and get a course of antibiotics, which would clear it up very, very quickly. And the last bacterial infection are the sort of food poisoning. So one of them is salmonella. This bacteria feeds on your food, and if it is not cooked properly, it doesn't die. It then goes into your guts and grows and breeds there. So it's important that you cook your food properly, and also once it's been cooked, you don't keep it warm for a long time. You put a lid on it, put it in the fridge, keep it very, very cold and refrigerated. You only keep it for a certain amount of time, and when you are going to eat it, you make sure it's heated all the way through to kill that bacteria. So the first group of pathogens are the bacteria. The next one is viruses. So viruses are not really living things that don't reproduce by themselves. They need other living things to allow them to reproduce. So we'll talk about the structure of viruses slightly later in the video, but these are a list of different viruses and their target organs. So Ebola used to kill a lot of people a few years ago, and that used to cause hemorrhaging, bleeding of the liver and kidneys, and that's how it would kill you. At the minute, the coronavirus pandemic is happening, and this causes a disease called COVID-19, which affects mainly the lungs and other parts of the respiratory system. The next one is HIV that you need to know. This attacks the immune system, and because of that, this can lead to secondary infections. So people that have HIV have lower levels of white blood cells, so they are more likely to get things such as TB and things like pneumonia. So bacterial infections that wouldn't normally affect healthy people can have a massive effect on people with HIV, and HIV causes another disease called AIDS. And people get AIDS once their immune system has been damaged to a certain point. The next viruses I just want to quickly go over are MMR. So you would hopefully have had your MMR vaccine or immunizations, and that stops you getting measles, mumps and rubella. The females hopefully would have had a HPV vaccine, and that stops you getting cervical cancer later in life. So HPV is a virus that can lead to people getting cervical cancer. Since we found that out, people have made a HPV vaccine, which will massively decrease the number of females getting cervical cancer. So that is an absolutely amazing thing that wasn't available when I was a child, but it's a recent development. So viruses not killed by antibiotics, but the vaccines will stop you getting viruses. The next type of pathogens are fungi. So fungi are eukaryotes. They have a nucleus, and mushrooms are fungi. Athlete's foot is one of the fungi diseases that you will need to know about, and it is caused by damp feet. So it's called athlete's foot because athletes had sweaty feet traditionally, and they were more likely to get this. If you've got athlete's foot, you can go to a chemist and get a powder which will keep the feet dry, and it also has a chemical in which kills the fungi. So athlete's foot is a human disease. Cholera dieback is a fungal infection that affects plants, so it affects ash trees. We'll talk about that again a little bit later. And the final type of pathogens are the protists. So the main one that you need to know about is malaria. It's not caused by a bacteria, a fungi, or a virus. It's caused by a single-celled animal. And this grows in your liver and it'll damage it if you've got malaria. And it's spread by a vector. So a vector is just something that spreads a disease. And the mosquito is the vector that spreads malaria. So a mosquito will suck somebody's blood, and it will take a little bit of that Plasmodium protist, and then when it bites somebody else, that protist will get into somebody else's blood and cause malaria in somebody else. So malaria mainly affects the kind of tropical countries. So that is quite a brief overview. I mean, it took 8 minutes, but that is, believe it or not, a brief overview of the pathogens. The next thing we're going to look at is cardiovascular disease. So I'll just mention that anything that's caused by a pathogen is a communicable disease, and that can spread. Things that cannot be spread, things like cancer and cardiovascular disease, they are non-communicable diseases. Also, any of the kind of vitamin deficiencies, which I'm not going to go into here, they are non-communicable. You can't catch them off somebody. But we're going to look at cardiovascular disease. So there's kind of 3 steps that you need to understand, and you need to understand the risk factors which are more likely to lead to having cardiovascular disease. So the first thing that needs to happen is damage to the artery walls. And this can be caused by chemicals that are found in smoke and tobacco, and also by stress. So having high blood pressure can basically stretch the walls and cause damage to the lining of the artery. What happens next is, normally the lining of the arteries are very smooth, and there's not very much friction between the red blood cells and the side of the arteries. It's normally very, very smooth, but now it's kind of being roughed up a bit. So what happens is things like fat that's carried in the blood naturally, we need fat, but what happens is it kind of sticks to that rough part of the wall. And if you have a very high fat diet, you are more likely to have a build-up of fat in your arteries. And what happens next is, so all that blood has to squeeze through a smaller area, so you have a really high blood pressure in this part of the vein, and this can cause further damage to this wall, and you get more fat building up. So now what we've got is a blood clot. So there is no blood getting through that artery. So this can cause things such as an angina. So people getting pains in the chest because basically the heart muscle isn't getting enough oxygen. And one other dangerous thing that can happen is this blood clot can actually become dislodged. It can basically get squeezed out. It can go upstream and it can lodge itself in a narrower artery. And this can cause things such as strokes if it affects an artery that is feeding the brain. And it can cause heart attacks if it is feeding an artery which should be feeding the heart muscle. So blood clots can move and they can be fatal, or they can also cause some life-changing things such as heart attacks, strokes. And it's important to note here that these things are risk factors. So the healthiest people in the world can get heart disease, but if you smoked tobacco, if you had a really stressful lifestyle, if you had a very high fat diet, you are more likely to get heart disease. And another risk factor is, do people in your family have it? Because there can be a genetic link as well. So again, I've kind of flown over the non-communicable diseases. This is quite a big topic, and I did kind of want to keep the video under 30 minutes long. So if you have time, if you want to really push yourself, you can go and look at things like vitamin deficiencies. Yeah, push yourself. But thanks a lot for watching anyway. I didn't want to waste too much of your time because I really want you to get to the end of the video and try some questions to make sure you really understand this stuff. Before we look at the virus life cycle, I just want to quickly look at the structure of a virus. So some viruses have got DNA, some have got RNA, but what they all have is some genetic material. This is surrounded by a protein coat, which is called a capsid. So the capsid protects the virus, it'll stop it drying out, and it can be very, very complicated. It basically has things that knock out the kind of defense mechanisms of its host, but it also keeps the genetic code inside of it safe. And all viruses also have antigens, and that allows it to attack a certain type of cell. So this is coronavirus, which is causing COVID-19. The antigens allow it to attack the lungs, but if this got into your liver, it wouldn't attack your liver because the antigens are wrong on it. But that is the basic structure of viruses. They're all different to each other, but they all have them three things in common. So now we're going to look at the life cycle. The first one we're going to look at is the one on the left here, the Lytic cycle. And lysis means to split. So what happens? Well, the virus has to attach to the host cell. So this diagram here is a virus which is infecting bacteria. If this is HIV, it is going to be a virus which is attacking the human immune system, the human white blood cells. If this is coronavirus, it's going to be attacking human lungs. So different viruses have different host cells, but this thing is the same in all of them. What it's going to do is the virus is going to inject its genetic material. It's then going to go into a circle, and circles of DNA are just called plasmids. That plasmid is then going to be read by the ribosomes in the host cell, and it is going to make new proteins by translation. So it's going to make new caspid, it's going to make new antigens, and it's going to also copy the genetic material of the virus. It is then going to assemble all that together to create thousands of copies of that virus, and then it's going to split open the host cell. And this is why it causes diseases. So if this is coronavirus, it is going to damage your lungs. It's going to make it hard for you to breathe because your lung cells have physically been split open. If this is HIV, it's going to attack your immune system because your white blood cells are physically getting torn apart. So lysis means to split, and that is the lytic cycle of the virus pathway. What some viruses can do, not all viruses, they can also have a lysogenic cycle. And what this means is it basically inserts the genes in splits. It basically inserts the genes into the host's DNA. So that circle of DNA from the lytic cycle can then insert into the host nucleus if it's a eukaryote. This is a bacteria, so it would just insert into the bacterial chromosome. And when the bacteria is going to split by mitosis, it's also going to copy the virus DNA. So this can happen hundreds, thousands of times, and it is going to proliferate in that population of bacteria. One human disease that can be caused by the lysogenic pathway are cold sores. So cold sores are caused by one of the herpes viruses, and to get a cold sore initially, you need to have kissed somebody that had a cold sore or basically been in contact with that virus. But once you get cold sores, they normally come up again and again at certain times of the year, or when you've got a cold, your immune system basically is fighting the cold, so the cold sore can then come out. But what's happening there is the virus inserts itself into your lip cells. The cold sore goes away, but as your lip cells are splitting by mitosis, the cold sore virus is in them healthy cells, and when your body is off fighting a common cold or something that you have, the virus sees its opportunity. That DNA will then come out of the nucleus, and it can then lead into the lytic pathway, causing a cold sore. So it will start attacking and destroying the healthy lip cells and cause a sore on your lips. So I'll clear that there so you can have another look and make some notes on the lytic pathway and the lysogenic pathway. Really important that you understand them two words. But I'll pause that there if you're not too confident. And if you are, we're then going to now look at plant defences. So different barriers that plants have to stop them being ate by pests. So lots of plants have really thick bark layers that have thorns or hairs which stop pests getting to the soft fleshy parts. And also lots of leaves have really thick waxy cuticle layers. So if you think about a holly leaf, it's really thick, and that is a waxy layer which stops pests getting to the soft insides of the leaf which the pests want to eat. Lots of plants have also developed poisons through evolution, and also insect repellents. So they'll give off chemicals which will physically repel insects and stop them being ate. So they are plant defences. Now we're going to look at different plant diseases. So why are humans interested in plant diseases? Well, it's all to do with farming and increasing yield. If a farmer has a field and has plants that are being attacked by pathogens or by pests, then this is really bad because it's going to basically lose the farmer money. It's also bad for mankind because there's going to be less food available for us. So what they will do is they will look at how basically the diseased plants are spread in that field. This can give lots of information. So if the wind is usually blowing to the left and the area that is being affected looks something like that, then this is probably going to be a fungus which is being spread by spores in the wind, or maybe a bacteria even. If it's spread now from a central point, this could be a pest which is breeding in one plant, or a bacteria breeding in one plant, and then it's getting to a certain number and then spreading to the plants immediately around that first plant. So distribution analysis can give ideas of what is causing the disease, but to know for sure you'll need a diagnosis. And what's often needed are lab tests. So you can have a look through a microscope at the pathogen and find out, is it a bacteria, is it a fungus? And knowing that can basically lead to a treatment, so you can increase the yield, so you can stop the disease affecting the rest of your crop. And another thing that would happen is soil analysis. Is the disease being caused by something in the soil? Is there a mineral deficiency such as magnesium? Is there something in the soil that is also stopping the plants growing correctly? So that's it for plant diseases, but please make sure you understand them words such as distribution analysis, diagnosis, and so on. It's really interesting stuff. If you'd like to go on BBC Bite Size or something and look at more really mind-blowing information on plant diseases, you can do, but I'm done with it. Thanks. So we humans also have physical and chemical barriers. So our physical barriers are skin, and we also have mucus lining the respiratory system. So bacteria that we breathe in will be trapped in mucus, and ciliated cells would waft it up from the lungs, and it goes into our mouth. We'll either swallow that, or we can spit it out or blow our nose. So skin and mucus are physical barriers. Chemical barriers include things such as hydrochloric acid, which is made in our stomachs and kills lots of pathogens that we ingest. Another chemical barrier are things called lysozymes. Lysozymes are made by our skin cells. They're also found in our tears, and they kill certain types of bacteria. They're basically antiseptic, so they are the physical and chemical barriers that you need to know about that humans have. The next thing we're going to look at is the immune system. So what happens if basically pathogens overcome the physical and chemical barriers and get into our bodies? So physical, chemical barriers stop things getting into our bodies, but what happens if that does happen, if things do get into our bodies? Right, a very brief overview of the immune system. So if a pathogen comes into our body, it will have an antigen on its surface. So all pathogens have antigens, and all white blood cells have antibodies. So antibodies attach to antigens. So what has to happen for the immune response to happen is a particular white blood cell with the particular antibody that's going to attach to that antigen has to be found. So this can take days. It can take, in coronavirus, this has taken like about 7 days, 10 days for this to happen. But when it does happen, that white blood cell is activated. And what it will then do is make lots and lots and lots of copies of itself, which can also, these copies can also make the correct antibody, which is going to fight off that particular pathogen. So some of the white blood cells, they can secrete antibodies into the blood, and these antibodies can basically work like little handcuffs. They can basically clump together the pathogens and by attaching them to each other. They can also label, they can basically stop the pathogen working. They can cover the antigens. So if this is a virus, it can't get into the host cell because it doesn't have antigens that can sense the host cell. A different type of white blood cell called a phagocyte can come along, and these cells are massive, and they basically surround clumps of bacteria or virus and engulf them. So they'll basically surround them. The cell membrane will move around that clump, and then it will release some enzymes into the gap in between, and it will kill and destroy the bacteria or virus that it is engulfed. So that is a very, very brief overview of the immune system. I will pause the video, make sure you understand them steps, but we're going to move on to the last two things, so antibiotics and monoclonal antibodies. Okay, so antibiotics only work on bacterial infections. They won't work for viruses or for fungus. But if you have a thing like a skin rash or a throat infection, you'll go to the doctors and they will give you antibiotics. If your infection doesn't go away, what they can do is they can send that sample off to the lab. They will grow that bacteria on a plate, and they will insert antibiotics on the plate. And what will happen, there will be a zone of inhibition around the antibiotics. So the bigger the zone of inhibition, the more effective that antibiotic is on that particular bacteria. So here, if you had this bacterial infection, this is the biggest zone of inhibition. So the doctor would then know, well, this antibiotic is going to work on that particular disease. So that's how antibiotics basically work. They can be put into creams or into tablets, and you make sure you do the full dose. So if you have 7 days of antibiotics, you need to make sure that you take all 7 days, because remember, if you don't, you'll kill most of the bacteria, but some of the bacteria has the genes that are going to survive. So when you have more of that antibiotic, the ones that survive are going to hang around for longer, and they're going to pass on their genes. So you will then get a strain of bacteria that is more resistant to that particular antibiotic. So make sure you take the full course of antibiotics. Right. Last thing we're going to look at is monoclonal antibodies. And monoclonal is very simply just a clone, a genetic copy of one of the white blood cells. So these are monoclonal antibodies. So remember, if you have a pathogen and there's one white blood cell that can fight that off, it's going to clone itself. And because it's cloned itself, all of these antibodies are exactly the same. So monoclonal antibodies looks like it's a really complicated term, but it just means antibodies that are identical to each other, and they are created by like one particular white blood cell. So one white blood cell that has made clones of itself, they would all make monoclonal antibodies. Right, this is where it does get pretty tricky. So if we need to make lots and lots of antibodies, how do we do that? So what we're going to do first is, why would we need to make lots and lots of antibodies? So in pregnancy tests, there's basically an antibody which attaches to a pregnancy hormone. So if you are pregnant, your body produces a pregnancy hormone which is found in urine. So if you weigh on this strip and you have that particular pregnancy hormone, the antibodies will stick to this part of the test and it will appear as a line. If you do not have the pregnancy hormone, then the antibodies are not going to stick to that line. So that will be clear for you. So this is one use of monoclonal antibodies. Another one in the future we'll talk about in a second, but we'll use that pregnancy hormone one as the basis of this. So we want to make lots and lots of the antibody that's going to attach to the pregnancy hormone. How do we make that? Right, what we could do is, we could just extract lots of white blood cells and they would make lots of antibodies. Unfortunately for us, that white blood cells, they're very, very hard to grow in the lab, and also they die after a certain amount of time. So what we can grow very, very easily in the lab are cancer cells. So these are immortal, which means that they would live forever. So they grow and grow and grow, and they won't die. So cancer cells are immortal. Now what we'll also need is a cell that's going to make these antibodies. So what we're going to do is, step by step, we inject a mouse with that pregnancy hormone. Bit harsh, but there you go. So we've injected the mouse with a pregnancy hormone. It is then going to have an immune response against that pregnancy hormone. It's never seen that before. It's going to find a white blood cell that attaches to that hormone, and it's going to make tons and tons of copies of that particular white blood cell. So what we're then going to do is, we're going to go in and we're going to extract a couple of them white blood cells. We're going to use a microscope and some really, really fine needles, and we're going to extract the nucleus from that cell. So we've got the nucleus from the white blood cell. We're going to take out the nucleus from the cancer cell, and we're going to combine them. So we've got the cancer cell with the nucleus of the white blood cell. What we can then do in the lab is we'll make lots and lots and lots of these, because they grow like wildfire, because they're basically cancer cells, they're immortal, and we can get tons and tons of these cells. So these cells are called hybridoma cells. Hybrid means more than one thing, and we can use these hybridoma cells to make tons and tons of the pregnancy antibody, and we can extract that antibody and put them into pregnancy tests and sell them for money. So in the future, what they think this might be useful for is for cancer treatments. So if tumour cells have got particular antigens on their surface, what we could do is we could create an antibody which senses that antigen, and we could attach poisons to that antibody, so it would only affect the tumour cells and not the other cells in the human body. So we could attach radiotherapy, like things that give out gamma rays. We could attach toxins to it, so chemotherapy to these antibodies, and that would increase the amount of toxins that the tumour cells are getting, and it would minimise the amount of toxins that the rest of the body would have. So side effects of chemotherapy is usually hair loss, because it's attacking your hair cells. So this would get over them sort of drawbacks of chemotherapy and radiotherapy, and it would hopefully revolutionise cancer treatments. So that's the whole revision for the triple B5 health and disease. I'm going to put up a question paper, a revision paper. Make sure you do that, please. Make sure you understand the different parts. If you don't, please email me. I will then, if you ask, I will email you the end of topic test for you to try. Thank you so much for getting to the end of the video. It is over half an hour, I know. I did want to record a video instead of doing this live, because I know people can stop at certain points that they don't understand. But yeah, thank you so much for watching. Stay safe, and good luck for the revision worksheet I'm going to send out, and also good luck for your end of topic test. Right, thanks.